Do you have any questions? How did you feel about the course? Is it gonna be interesting? Is it gonna be more difficult than you thought? Is it gonna be less difficult than you thought? Anybody wants to comment?
It seems really interesting.
Okay, thank you. New stuff. Okay, so a little more difficult than I thought, but interesting. Okay, the idea is that I don't want to make the course difficult for you. The course should be easy because first of all, if it's elective, and then it's a practical course. So that's why I want to keep discussing with you after each couple of lectures, because I don't expect you to go to a lab and run the experiment right away. Yeah, so it will be a lot of new things, and it's important for you to know new things because I guess molecular genetics, molecular biology has discussed many of the techniques that I'm telling you, but probably they didn't go in depth for their applications and how you will use in your everyday lab setup or in your job. So that's why it's important to touch on those things. Now, I try also to add a lot of videos for you because that's also a very important part. Because when you want to see how to do something, you probably want to see someone has done it before and you see the steps. Like if I say wash the cells, what does it mean wash the cells? Like when you see how you do it, it's just simply drop some phosphate buffered saline into the cells and then take it out. That's how you wash the cells. But the name looks really crazy. Okay. So I'm gonna share my screen. Please let me know if you see it so that we can proceed. The recording is on. Okay. Okay, so I will add up on the cell culture in health biosciences. So we discussed a very brief introduction last time. We just touched on the first step on how to perform cell culture and how we can get a cell culture. So I'm not going to go again into all the details on the first couple of slides, but then we will go more on the fact that we are trying to use these cell cultures for a reason. The most important reason is that they are cheap, they are available, and they are like a small microenvironment where we can test the drugs, where we can make a model system, we can generate viruses, we can engineer some parts of tissues, and we can use it for a model for cells that has been mutated or that acquired a mutation or they have a mutation. This is like gene or stem cell therapy. You can use it as a model for studying the cancerous growth, the tumor growth, as well as to test some biohazards that we don't, like drugs that you have achieved, biohazard chemicals and their effects on different systems, on different cell types. So that's a very, very good and powerful system. And it's very cheap, as I said. Many labs can run only cell culture and they can perform a lot of research on that. When the research go to a second level, probably it's not only cell culture, it will become a more complicated cell culture. And I will show you on the last 3 slides today how we move from a conventional simple cell culture into a 3-dimensional cell culture and organoids and spheroids and how those can be used. Now, eventually, when you do all your experiments in cells, what you would like to do is to test it in an animal model. And that's where zebrafish, mouse, rat, pigs come into the picture. Okay, so we discussed the difference between cell cultures based on their function, based on their appearance, based on their ability to detach or not on the culture plates. And we discussed that, of course, every cell we get into the lab, it comes from a particular organ, and therefore an epithelial cell is different from an endothelial cell based on the environment where the epithelial cell was and the basement membrane where it was attached compared to the endothelial cell and all the growth requirements. Therefore, you can imagine that an epithelial cell would need a different growth medium than an endothelial cells. And a fibroblast as well, like these three could be that the fibroblast grow in both endothelial and epithelial cell, but endothelial cell cannot grow in that medium. Given the difference in growth requirement and given the difference in the basement membrane where they were. So it's important for you if you get to a lab and somebody tells, okay, the technician would tell you probably you're going to use this cell type. It's important to directly read and know what is this cell type, where does it come from? It's an epithelial cell, it's an endothelial cell, it's a fibroblast, it's a mesenchymal cell, it's a neuronal cell. So that's very, very essential to know before you propagate in your research. And as I go in the slides, you will see that this adds up more and more. We discussed, for example, the process of the loss of differentiation and its role in the capacity in culture. So the more the cells get specialized, the more difficult it will be to culture them, because the components would be like the growth medium will be very complicated. There might be one factor that is missing in your growth media, and the cells will not be able to grow. So if I ask you a question right now, based on this scheme, which cell is easier for you to grow in culture? Would it be an insulin producing cell or a fibroblast? I would like somebody to answer. Fibroblast. Good answer. So fibroblasts, because they are not specialized. And I particularly chose fibroblasts because it's known to grow, it's the best cell to grow anywhere. Like the growth medium requirement and the culture plating, the surfaces, the fibroblast can grow anywhere. However, as I said, the insulin producing cell, it's a more specialized cell. Therefore, the growth requirement for that cell is different, and the contact with other cells is different. And that makes it more difficult to grow in culture. And you can imagine this applies on many other cells. If I tell you, for example, brain cells, they will be more difficult to grow than fibroblasts. And the same if you take a heart, you have cardiomyocytes, you have fibroblasts there. Cardiomyocytes are so difficult to grow compared to the fibroblasts. Okay, Riley, so I have a question. You can unmute yourself, yes.
Oh, great. So I was just wondering if there's any type of specialized cell that that kind of goes beyond the conventional approach of the more specialized, the less chances of you having a successful growth. There's some that from your experience or just general knowledge that would kind of show, hey, there's some specialized cells that may actually be able to be easy to grow.
It's generally acceptable that the more specialized the cell is, the more difficult to grow it in culture. And this is probably, I can give you one reason behind that. Some of those specialized cells, for example, they cannot divide. Like if you take cardiomyocytes, they cannot divide. So in principle, if you dissociate a heart and it has like 3 million cells in that heart, 3 million cardiomyocytes, and you put them in a culture dish, they are terminal in their division capabilities. So they are not going to divide.
So from that understanding, it's just very important to keep your cells
Yeah,
alive. If that's the case, when you're studying the specialized cells, they're
absolutely.
probably in high demand.
Absolutely. So
Okay.
it's important to keep them in the suitable environment and the suitable growth, because they are very limited and they are so precious. So that's a very important thing. And in this case, what I gave you, you are not going to get, if you keep them in culture for 1 week, they are not going to divide. So you only have the exact number you started with. It's important to plan your experiment or whatever test you want to do, a drug or whatever reason you were using those cells. It's important to plan it timely, that you do it within this week. Otherwise, you're going to lose your cells because they can't stay forever.
Thank you. Thank you.
Oh, you're welcome. Okay, so based on this, we discussed the last time the process of acquiring how to prepare cell culture. And it says, this slide was, I looked back and it looked a little bit confusing, and I added some more associated slides to make it easy for you. So please, if you raise your hand and I didn't see you, just unmute yourself. You're welcome to talk and I will come back to you. So in this slide, it says we will resect like a tissue, and then we will do a culture out of it. And across the different steps we follow up, it will end up, we will generate a cell line, and we will immortalize cell line. So this means the cells will be able to grow forever in principle. So I have done this before. I published last year a paper about this. So I have it added to your module so you can quickly check how I generate fibroblast from heart. It's very simple. You will take the heart, you will digest it, and then you will put it in a growth media, add it in culture dishes, and that would be the cell culture in vitro. That's the first step here. And then when the cells in the dish, they become 100% or 90% confluent, meaning they fill the space and there's no physical space for them to grow, then you want to split them. And that's what we call the subculture or the passage number 1. And if you repeat this again, this will be another subculture. So every step you do, it's called a subculture. And normally in a lab, when we take the cells from the first time from a tissue, then we do the first primary culture. We call this passage number 1, meaning the original cells has been passed one time from one plate to another plate. And we keep recording this all the time because we would like to know how many passages were done. Okay, I'm going to stop to answer a question for your colleague. I can't see you raising your hand anymore.
Hi, can you hear me?
Yeah, please go
Yeah,
ahead.
perfect. So with respect to the definition there where it says a primary culture is subcultured or passaged, the diagram shows that the cell line is derived from the secondary culture. So
Mhm.
would it be fair to say that like the subculturing procedure must occur multiple times in order to derive a cell line? Is that
Yeah,
like a more accurate definition?
yeah,
Okay.
yeah. But this is from a practical point of view, because
Okay.
if, for example, you digested a tissue and you brought the cells out, which is in step number 2 here, you have limited number of cells. You want these cells to divide and grow in order to have a backup. And I will explain that there is a backup that should be done on all these cells. Like imagine in step 2, you have one plate. Then in step 3, which is the primary culture, probably you will get 2 plates. And in step 4, probably you will get 4 plates. Now you can take these 3 plates and probably freeze them and continue working on one. And therefore, you don't need to go to the tissue again to generate the cells. So this is very important. So this, from the point of view that you have to passage them, yes, because you need to increase the number of the cells you have. And at the same time, probably you don't want to go from 0 every time. So to have a backup. Okay?
Awesome, thank you.
You're welcome. Okay, so then we spoke about the cell line immortalization, and we spoke that the cell line immortalization, now you are moving more toward having a more homogeneous type of culture. Remember, when you take a tissue and you digest it, you have all the cell types there. Probably you can eliminate some because some are bigger than others. Like imagine there's a muscle and you are digesting a muscle. The muscle cell is really big. Fibroblast endothelial cell are small. So by centrifugation, you can separate. But then the interstitial cells, normally the fibroblast, the endothelial cells, the leukocytes, they are similar size. So probably at this point, you cannot differentiate between them. They are all together in the cell culture. But what is defining which cell is growing is the culture media and the attachment situation. And those I will describe in the next couple of slides. Now, the immortalization process we described last lecture, but I added for you. Okay. So this we already discussed as well. So I added for you some of the definitions that you have to remember about the cell culture. So what's the definition of an adherent cell compared to a cell in suspension? So in principle, those cells are able to stick to a culture dish, while cells in suspension, they just grow. They don't depend on an anchorage to grow. Immortalization, and what's the process of immortalization? I also added for you here a video about how you achieve immortalization. It's around 6 minutes. It gives you a good background and a summary of what I discussed last time about the usage of these viruses, the SV40 and the telomerase reverse transcriptase protein, and the upregulation of the RAS and the MYC, as well as the downregulation of the tumor suppressor genes P53 and RB. So now the immortalization process that is achieved by the usage of the virus, it would look like a little bit difficult for you because you don't know how viruses work for now, and we didn't discuss this yet. But in 2 lectures from now, we will be describing the lenti and adenoviruses, and this will be very clear. So we'll just go and watch the video to see how we achieve the immortalization. And if you have any questions, come back to me in the next lecture. I also added for you this PDF file here embedded in the slides. You can go to the link and download this PDF. It's like 3 pages or more. And it describes for you step by step those immortalization process. But the bottom line, I described that for you. It's a very simple thing. You overexpress one of these genes, the RAS or the MYC or the TRT, or you downregulate the P53 and RB. So that's the ultimate goal of the immortalization process. And that saves the telomeres of those cells, because every time we do the passaging, the telomeres of these cells start to shorten, and we lose some of the genes for the progression. And therefore, if we maintain the telomeres, the cells will stay in culture for longer time, and that's what exactly happens. Now, with immortalization, it's important for you to know that there are some changes that happen to the cell line. It doesn't look like as the cell line as the cell that you took from the original tissue. So if it's a skin tissue or it's an epithelial cell from, let's say, intestine or a kidney, when you do the immortalization, that's a virus. The virus gonna change the cell sometime. It will maintain a lot of original properties, but it also loses some of the properties of that cell. So it's always important whenever you use a cell line in culture or an experiment in a lab you go back and double check it in an organ, like in a setup in a mouse, or you take it from a human directly if you can and test it there before immortalization, because some changes are due to the immortalization process, okay? Now, what are the basic requirements for the cell culture? And these are important for you if you get into a lab. You will get into an environment where probably some of the machines you never seen and some of the media and the components and the protocols you have never seen. So the growth environment is a very essential point for the cell culture. And the growth environment is not something that we know, like per se, like there's a lot of research about what are the growth environments that are needed for different cell types. And every other year we have an update on, oh, this cell culture is growing better, for example, in this media where there's some supplements added. So this is something that you cannot control if you go to a lab. They will provide you with the cell line, they will provide you with the growth material, and they will say, this is what we are using for our research, and therefore you have to use it. It's always good to keep an eye on the research that is coming out to tell you that probably this cell line has been cultured in this situation, and we have achieved much better results. Then you might want to change your culture condition to that one. However, if you have performed like 2 years of research using a growth environment, you don't want to suddenly change it and go to another one just because the cells are growing better. Maybe you want to finish that research, and then when you start another round of projects, you make these adaptations. So the solid support for the cells is very important because this is where they grow in case they are adherent. So they have to attach to a plate or a matrigel, which is like a scaffold where the cells grow. In conditions where it's a suspension, they don't care about the anchorage and the adherence, but they do really care a lot about the growth environment. The growth medium should support with nutrients, of course, the sugar, the proteins that are not produced in a cell, a lot of ions like calcium, magnesium. It should be the right buffer. It should be also supplemented with vitamins and growth factors. So all those, when you buy growth media, you will find within. Now, the solid support for the matrigel, it's not something you would do unless you already experienced the good cell culture in a plate or in a flask. When I say a plate till the end of this session, this means cells adhere to the plate. And when I say a flask, this means the cells are in suspension, so they are just flying around. And the best example for you is what we call by the commercial name. So if you get to any lab, they will tell you, we have HEC293 cells, and we grow them in DMEM. So the name is written here, DMEM. Now, don't ask me what DMEM. I honestly don't remember what it is. So DMEM is simply a growth media that is sold by a company called Invitrogen. It contains a lot of all the material, the growth requirements for cell types, and it could be used for multiple cell types. For example, the 293 cells that I said, they are called HEC, human embryonic kidney. 293 is just because the clone was 293. They can grow in DMEM. Fibroblasts as well, they grow in DMEM. So if you look on the bottle, they will tell you what exactly it has in terms of amino acids, in terms of the vitamins, the amount of sugar, and the electrolytes and the ions. Now, on top of the growth media, you have to supplement the cells with, this is a very, very important, you have to supplement the cells by serum. And in 90% of the cultures that I did in my life, we use 10% serum. This 10% serum, this means 90% of the growth media that we buy that contains these material that I don't really, I'm not going to ask you what are the components, of course, and 10% of the serum. Now, the serum can come from bovine, like this is from the cow, or from the calf. It depends, like based on the age, because if you take the serum from a younger animal, it's different from an older animal. So the serum you use is important to also know. You have to be very careful to make note about the lot number of your growth media, because from one lot to another lot from the company which you are buying, the components might change a little bit, and that can change your culture. The same thing, the reference number, of course, is very important beside the lot number. And the same thing go for the serum. Now, if you provide the cells only growth media, they will not grow. They must have serum on board. Okay, so if you get to any lab tomorrow, oops, let's go back. If you go to any lab tomorrow, so maybe I have to do this better. Okay, if you go to any lab tomorrow, then you will find few equipments that are important for cell culture. This is a cell culture incubator, which you can see it's like, it looks like a fridge, I would say, but it's 37 degree. This can be sterilized internally with UV light or self-washing. You will see a laminar flow hood where you can perform all these culture steps under sterile conditions. You can have probably a UV lamp under those hoods, so that after you're done, you can turn it on for 1 minute or 2, so it can sterilize the environment. And probably if you're doing this on the bench and you can't do this within the hood, you can use these burners to stay around the fire in order to maintain the sterility. So in principle, you would need a stable environment and a sterile environment. So what does it mean, a stable environment? Meaning you are taking a cell that is growing within a body that has an oxygen content of 21% and carbon dioxide of less than 5. So you want to maintain this within the incubator. Of course, you will need humidity that gives the water component so that the cells don't dry out and the cultures don't dry out. Otherwise, you will put, if you don't provide that humidity, you could put your culture media and cells in that incubator, and you will come back and you will see it dry. So nowadays, these incubators are equipped with systems that can tell you CO2, oxygen, humidity, and temperature. Anything drops or go beyond, it start beeping, so you will know and run directly to the lab to see what's going on. Now, the sterility, as I said, it's achieved by UV light. You have to wipe your hands, use gloves all the time, wipe all the surfaces that you use, and avoid touching anything during doing the culture, like your phone or the handles of doors. Then you should, with all these measures, you still get contaminations. So to avoid contaminations, we add antibiotics and antimycotics, which is antifungal and antibacterial. We add it to the growth media. So at this step, you have your growth media that you bought from the company. You have the serum that is added to the bottle. Let's say it's 500 ml normally. So you need 10% serum, let's say. You add 50 ml of serum on top of this, because 10% of 500 is 50. And then probably you want to add 1% of antibiotics, antimycotics. So you will add like 5 ml of penicillin, streptomycin, gentamicin. There are those you can buy in mixtures and you can add them to your growth culture. And now you have a bottle from this that is ready to use. You already labeled it. This is DMEM, let's say. it contains 10% serum, and it has 1% pen strip, and therefore you are ready to perform your cell culture. So the growth media is great. Now, let's say you arrive to a lab and somebody say, okay, this is the cell culture cell type we're using. This is the culture media we're gonna use, and here's a plate I already prepared for you. The first thing you have to think about is that within this culture, even though we did all those measures, probably we will have a contamination that we cannot see. And in culture media, and people who perform experiments, they know that routinely they have to do tests for the bacteria that's called mycoplasma. So the characteristic of this bacteria, it's this wall-free bacteria that's insensitive to antibiotics. So even if you use this mixture of antibiotic, this mycoplasma is still able to grow. And of course, if a bacteria is growing with a mammalian cell, the environment is going to be not normal for the mammalian cell, and therefore the physiology of that cell is going to change. So you might be in a situation where you have done an experiment and you got the results on a drug testing or DNA mutation or signaling molecule, and suddenly this is stopped. You cannot find it anymore. The first thing you have to ask yourself is my culture contaminated with mycoplasma? Because this is not going to appear under the microscope, and this is not going to be visible in the culture. In order to see the mycoplasma, the first thing you might want to do is to put a stain for the DNA. And if you can see in this image here, these are mammalian cells. It looks very nice nuclei. And you see a lot of very, very detached small blue staining here. So the DAPI stain is very particular for the DNA. So therefore, the nucleus turns blue under UV light. And then in the culture media, you see a lot of these, and that's a typical situation. This is the DNA that belongs to the mycoplasma. So this is very important to keep in mind. Also, there are ways to test this. You can take culture media and you do a PCR on it, and you can detect the DNA specific for the mycoplasma. Okay, so you have the growth medium. Now we want to, let's say, grow the cells. And in order to grow the cells, I'm putting for you here an example for adherent cells, because this is very, very important to follow up. In order for the cells to adhere, the plastics that I showed you in the previous slides, they should be modified. So in other words, if you get into a lab and there are two packs of plates that are exactly the same, one of them should be not treated, let's say, and the other one is treated, and you want to grow your culture, you have to be very careful which pack of plates you will use. Obviously, the companies which produce these plastics for cell culture, they have worked years and years to make the plastics suitable for growth. And one thing that those companies do is to hit the plates with ionized gas, and they call it corona treatment, so that the polystyrene, which is the material that is used for the plastic, becomes charged and suitable for cells to adhere. Okay, now one company to another, like there's like 4 or 5 big companies who do that. Now we have many sub companies, but the modifications can be summarized as you see in this image here. They could hit with the gas, and this could lead to the roughing of the surface. So instead of being like very smooth, it will become with dentation. It could be that when you hit, you have to also add positive charges or negative charges, or both positive and negative charges. And you can also add some polar groups, that is like some sort of additions that contain multiple of positives and negative, but the final outcome is either positive or negative. So in this situation, this comes empirically, like companies tested negative charging, positive and negative or polar, and they came into market with some products where the cells can grow better in these situations compared to polystyrene only. So therefore, if you are growing a cell type, as I said, first, what's the cell name, where it comes from, which tissue, and then what growth media people are using. 3, serum, and of course, antibiotics to avoid contamination. And now the plastic plates, they should be treated. So you have to be careful that your plastic is treated in order to propagate the cells. Otherwise, it happened before with many people I've seen, they grow cells in plates that are suitable for bacteria, and suddenly all the cells detach the day after, and all the cells die. Now, on top of the modifications that the companies do when they send us the plates, there should be some other modifications that we learn from the cell culture conditions through research. So in other words, if you take fibroblasts, as I said, and you throw them on a plate that we just bought from the company with those modifications, the fibroblast is going to attach because it has a lot of capabilities to attach to surfaces and to crawl. While if you take endothelial cells, probably they will never do that. Or you take muscle cells or cardiomyocytes, they will never do that because these cells, they don't have the capabilities that a fibroblast has. Therefore, on top of having the plastic plate prepared from the company, we need to add some coating to that plate. In other words, I would take that plate and I would add like 3 or 4 ml of this material, and I added for you a list here. For example, polylysine, this is an amino acid with the L or the D format, conformation, on ornithines. So this will just make a cushion for the cells to attach. There are multiple ways. So this is like polylysine, polyornithine, gelatin. Gelatin is one of the most used because it's very cheap and it's available all over. Now, depends on the cell type, and I go back to the question that your colleague asked me. Maybe cancer cells can grow in most of the environments. Cardiomyocytes, probably they need laminin. Fibroblasts might need collagen. Specialized fibroblasts might need fibronectin. Like this depends on the type of research you're doing and the cell type that people are using in research. So this is not something you will invent. You will go back to literature and see what other people have used, and you will simply reuse it. Now, this is in very simple environment, which is the two dimension. If we go to 3 dimensions, sorry, the collagen gels and the matrigels are the most used now. As an example, the collagen gels are used for contraction assays. For example, skin contraction. And if you are using the matrigels that go for cancerous cells, and most of the time when they do a tumor environment grafting, the matrigels are used. So down the road, when we discuss the animal models, I will describe for you what is the matrigel. And just give me a second because I received like 7 different emails about what. I just want to see if some of your colleagues are not able to log. Okay, so it's not related. Okay, sounds good. I would like to stop for a minute here, and if you have any question or speculation, just let me know, and then I carry on. Any questions? Mhm. Okay. Well, I didn't see any question, but I want to make sure. Let me see this chat. On slide 15, or first about the modification, the modification positive charge, negative charge are dependent on what you are culturing, right? Well, the most of these modifications, to be honest with you, we might not know, because when the company send you the culture plates, probably they would not tell you what is the treatment. We would know that they are charged, and the way it works, when you are culturing a cell type, the best thing you do is you contact the company which provide these plastics, and you would say, I'm culturing, let's say, 293 hex cells. What is the best culturing dishes that you are using nowadays? And they will send you a copy. Probably during this communication, they will give you some information, but on the plate itself, they will say it's treated. It's most of the time is not written. Okay, so anytime we're immortalized a sample, we're making a virus that is slightly different from the original cells. Wait, so Kanwar, you had a question before that I didn't answer. So when you are creating immortalized cells, you are using the virus, which is exact same virus, and you are throwing it on that cells. And what the virus will do is gonna give a better capacity for this cell to divide more and more and maintain the telomeres. And this is achieved by knocking down, bringing down the expression of p53 and RB, or overexpression of MYC and RAS and the TRT. So that's exactly what you do every time. Now, when the cell is infected with the virus, after a few days, it will become more capable of division, and it will become really proliferating for a long period of time. Okay. Okay, sounds good. So now, so that's an example for you. And if you look on the slides, there's a lot of more references for you. Not necessarily that you need to know everything on those references, but I'm first, for ethical point of view, I have to provide references for the slides, and then for those who would like to read more, they can go to these and read. But in principle, I'm giving you the conclusion of that on the slides. So you can see there's an example here where the cells were plated on fibronectin gelatin mix or not, and you can see the difference between the two. Already, if you don't code this, the cells will be dying and not attaching. And if you look in a culture, well, probably the cells in the original culture are not great. The healthy cell, this is, I mean, if you can see on the screen with a good resolution right now, this is a good cell because it's attached and it has a spindle-like shape. Like this is a good cell here. This is a good cell. That these are in a very good condition. But if you look on the cells that are rounded like this, those are not healthy already to start with. But overall, if you compare fibronectin to the no coating, there's a massive difference on the culture efficiency. Now, in your cell culture, if the cell is dying, if the cells are adherent and they are not in a good situation and they are not healthy, you directly see that they are rounding up. So the rounding up in your condition, in your culture, is the first indication that your culture is not in a very good situation. Okay, so now we have the cell line, for example, that we are going to use. We have the culture media that we need, and now we have the plate or the flask that we will use. We checked the cell type, we checked the growth media if it has, how much need of serum. We checked our incubators, if it needs 5% CO2 or 10% CO2. We have the plates, we check that they are treated by the company, and then I looked on the literature and I've seen if the cell would need an additional coating like the one we describe here with gelatin or fibronectin or collagen. So all that is set up and my plates are ready. And now I grow the cells. So we grow the cells, and let's say we had 1 million cells grown on this culture plate. This is like 10 centimeter, let's say, in diameter. And after 2-3 days, I come back, I check in the incubator, and the cells are almost 80% confluent. So this means the spaces are almost gonna be filled. So I need to split this cell. So this is the first passage. So the passaging is required. When is that going to be required? When the cells become confluent or when they enter the stationary phase. And I will describe the kinetics of the cell culture in the next couple of slides. So the procedure in order to change the passage or to subculture those cells is, first, if they are adherent, I have to lift them from the plate. Meaning I have to use an enzyme, that's trypsin, in order to detach the cells from the attached part on the plastic. If the cells are growing in flasks and they are in suspension, you don't need this step because they are already in suspension. All you need to do is to do centrifugation. So if we continue just with adherent cells, so we detach them with trypsin, centrifuge and resuspend and put in a new. So if you have 1 million cells here, probably you're going to take it and you divide it into, let's say, 500,000 each or 200,000 each, depends on the experiment. That's something else. Counting is something, and knowing how much cells to add there for your experiment is something else. Okay? Now, the steps is when to do that. We know it. Now, why we should do that? First, to avoid starvation, because the more cells you have in the same plate, the more they are using that amount of volume of growth and growth nutrients in the environment. And therefore, after a while, you get depleted from those nutrients. You will not have enough for the cells because there's too much cells. So as an example, this culture plate that you see is like 10 centimeter in diameter. We add 1 ml per centimeter. So it's like 10 ml of media. And therefore it stays up to 3 days maximum. So you might want every 3 days to change the media on your cells. The other thing is that when the number of cells increase, probably the growth is going to stop because the cells, they don't have physically a space to grow. And the growth is inhibited by something called contact inhibition. So the more the cells are squeezed around each other, the more signaling they will tell that don't divide. So this is called contact inhibition. Contact inhibition, okay? So the passaging process is that we will wait, this is 80% around, and then we will wash the cells. So we will simply suck out the culture media. The cells will stay attached because they are very well attached to the plate. You have to be careful that the magnitude of attachment is different. Therefore, if the cells are like slightly attached, and if you do some physical movement, you can take the cells out. So you have to be kind of gentle. You suck out the cells. Now you suck out the growth media, and you end up with the cells only in the plate. You add phosphate buffered saline, which is just saline, just to wash the cells. If there's any dead cells or any components from the growth media you don't want anymore, you take it out. Then you will add around 3 to 5 ml of trypsin, and trypsin is going to make the detachment of the cells. The contact with the plastic is going to be destroyed, digested by trypsin. You have to be careful that you don't leave them more than 5 minutes with trypsin, otherwise trypsin starts killing the cells. And the amount you have to keep on trypsin, it depends from one cell type to another. You have to quickly run back and forth under the microscope to check. And an example I gave you here, 293 cells, they detach very easily. If you add trypsin or if you like hit on the plate on the side, some cells detach. Okay, and then after a few minutes, let's say after 5 minutes of adding trypsin to the plate, you will collect all that environment. Like let's say if you added 5 ml of trypsin, you add another 5 ml of phosphate buffer or the growth media, and you wash and you collect this out, and now the cells are in suspension. And you can put them in a tube and you can centrifuge and you pellet the cells. And if you do this process, you would need minimum 2 hours, some cells 3 hours to 4 hours to reattach again. Some cells would need like 12 hours to attach again. So you have to be careful the cell type, you read more about it, but that's the general procedure. So from one original culture, we have a subculturing, and that's the passaging process. And we keep doing this. Like if you are doing an experiment, you count the number of cells and you put the corresponding number of cells in the plates, and you can treat them with a drug or hit them with a situation where you starve them with no serum. Depends what you're doing. That's another thing. But also imagine that you want to preserve the cells. Like now I have the second subculturing, and maybe I don't want to do experiments. I want to go on vacation, or I want to send it to someone other on the other side of the globe. You want to use my cells to do experiments. What can I do? So that's why we do cryopreservation. And I will describe this in the next couple of slides. Now, just before stopping on the cryopreservation, I want to describe for you the plating process and the passaging process in terms of the kinetics of the cells. And this probably is applicable on any cell culture you do, not only mammalian cells. Like if you do yeasts or bacteria or mammalian cells, I think they all follow the same type of curve. Probably the timing is different and the amount of media and the conditions are different because each has a different growth media, different number of doublings and all that. But the steps of the growth curve is the same, where you would normally start with a lag phase where you added your cells. Like today in the morning, you counted like 100,000 cells or 500,000 cells, and you put them in a plate of like 2 centimeter diameter. Those cells are not producing anything in terms of the growth. So their number is not going to change within like 2 hours. At least you have to wait 24 to 48 hours to start having changes in the growth. So that's the lag phase, right? Then when the cells kick off and start dividing, that's where we see the exponential growth phase. And in both conditions of the lag and the exponential phase, there's the common thing that the cells are healthy, they have enough nutrients, they are fairly like 90%, 99% of the cells alive. So this continues for hours and hours and till the end of the growth phase, the exponential growth phase. And that's when we start reaching the stationary phase, where because of the lack of the nutrient or because of the physical contact inhibition, the cells start to die. Some of them, of course, start to die, but the majority of the rest of the cells are still alive. So when you reach this stationary phase, ideally, you want to split your cells. So you have to passage them. Otherwise, if you wait long, like if I give you an example, NIH3T3 cells, which is a fibroblast cell line, the name comes a 3T3, meaning you have to do 3 passages per week, meaning you passage, let's say Monday, Wednesday, Friday. If you don't passage them 3 times, if you pass them Monday and you wait and you passage them on Friday, they will be super confluent and they will be mostly dying. So this is, for example, a cell line that people know and use a lot in the lab. So the stationary phase can be known, that's what I want to say, for each cell line. If you go back to people's work, they will say we are using HEC293 cells, we split them every 72 hours. So every 72 hours, you have to keep doing that. Others, they would say every 10 days to grow. Depends on the cell type. That's something you have to read based on the cell type and the growth condition. Now, if you leave them, as I said, long on the stationary phase, they start dying, and the majority will become dead. And if you eventually don't subculture them here, they will eventually die. Okay? Now, it's very important for you to know why I'm telling you this, because if you want to do an experiment, it's very important for you to maintain the cells within the exponential position here, exponential growth. Otherwise, imagine you are trying to see if the drug kills the cells or not, and you are using the cells in this phase here. You already have 70% death in your condition, so your experiment is not going to work. Similarly, if you use cells at the end phase of the stationary phase, right? You already have a lot of dead cells. So you have a lot, like 10 to 20% background of your death that doesn't come from the drug. So you have to be very careful to maintain the cells and eventually keep them in their exponential growth phase when you do experiments. And these conditions, as I said, depending on the cell type, the type of research, and the timing. I gave many examples and I said 48 hours. Sometimes you want to do a test only 5 minutes. You don't need to do a test for 48. Like if you are adding a cytokine like interleukin B or interleukin 6, it can trigger a signaling on the cells within like 5 minutes. So maybe you want to just add it 5 minutes and collect the cells right after. In other words, the experimental setup is something else. But the condition of the culture is the most important, that the cells should be in their exponential phase to get you the results that you are expecting. Okay. Let's stop for a minute here. And if you have any questions, please unmute yourself and you can ask. Otherwise, I carry on.
Could you just explain one more time the lag phase and what's happening with the cells?
Yeah. Okay. So the lag phase is normally the phase where you either just took the cells and plated them, or you just acquired them out of a tissue, or let's say from let's say from blood. Somebody donated blood and you are using blood cells for your condition. You just have, let's say, 100,000 cells that you obtained from a donor. At this stage, you are not able to do the experiment because you have a limited number of the cells. Normally,
Okay, so I think I'm just struggling. Is this lag phase like an incubation phase, or is just a phase of you just putting it on the dish, waiting a few moments, and then putting in your covers and stuff?
Normally the lag phase is either you don't have enough cells, you need them to grow to become more. And that's, for example, you add the cells today and you have them tomorrow, exact number that you need. Or the cells need the time to attach. As I said, some cells need 2 hours, some cells need 4 hours, some cells need 6 hours to attach very well. So this is a lag phase also.
That's perfect for me. Thank
right?
you.
And there's another thing also in the lag phase. The attachment probably dictates what you can do next. Because imagine you want to do a drug that you want to add only for 2 minutes, and then you want to take it out from the media. You first have to plate the cells and make sure the plates are, the cells are attached. And then, because if you add the drug 2 minutes and you suck out the media, you're going to suck out the cells with it, right? So you have to be careful on that condition. So the lag phase can come from different, from the number of cells, from the condition of the cell, or the treatment you want to do. It can cause that, right?
Yeah, thank you.
You're welcome. Any other question? Okay. Please unmute yourself because that's much easier, and we can like discuss by chatting rather than writing. Okay, so as I said, imagine you want to generate a cell line, and then you can't be in the lab day and night for 3 years continuously, because if you stop using the cells and they stay 48 or 96 hours, they will die. So that's why scientists came with the cryopreservation. And in fact, cryopreservation is something that we know from paleontology and the study of the frozen samples. So the cryopreservation is a special way that we use in order to preserve cells, in order to revive them later on for whatever reason. And essentially what we do is just we block the metabolic and the enzymatic activity of the cell, so that when we come back and we put it back in culture, these conditions, in a very favorable condition, these cells will regrow, and then we are able to perform whatever experiments or tests we want to do. Okay. Now, the cryopreservation is something very, very important, because it's the initiative for bringing different cell types into preservation, like the blood bank for the blood cells, the sperm for preservation of sperm bank, and all the different cell lines that people use now in research, in addition to organ preservation. And that applies also on plants for seed banks. So when we do the cryopreservation, we cause damage for the cells. So in order to avoid the cryo damage, we have to use what we call the cryopreservatives. So the cryo damage, just to tell you, as described in this scheme here, if you fast cool the cells, and normally cryopreservation is happening at -80 degrees in liquid nitrogen. So if you compress nitrogen, and this is the container that you see here, if you compress nitrogen, it becomes liquid at -80 degrees from the gas condition to the liquid condition. And in these containers, they have a very high pressure, of course. If you add your sample there, they will be maintained at -80 degrees as much as liquid nitrogen is there. And that's why liquid nitrogen has to be replenished every other week. Now, if you fast cool the cell from 37 degrees to -80, this will cause cell death, because there will be crystal formation within the cell that will make rupture of the cell membrane. Now, if you do a slow cooling, instead of going like only 2 degrees per minute or 1 degree per minute to drop, this will not cause the crystal formation. However, the water within the cell is going to be transported outside, and this will cause the cell dehydration and collapse. And in both situations, the cell will die. And that's why people use the cryopreservation in order to prevent the formation of the crystals and to keep the water within the cell, so avoid the transport of the water outside of the cell. And that's how we achieve to have a slow with a cryopreservation that leads to the preservation of the cell up in -80. Now, there are many of the components that has been tried, the glycerol, ethylene glycerol, propylene glycol. But the most used nowadays is the DMSO, which is the dimethyl sulfoxide. If you go to any lab, you will hear about this. You add 10% of this on any cells, and you directly put on those Mr. Frosty thing. And the Mr. Frosty thing, you put it in the -80 degrees fridge, and then it will drop the temperature 1 minute every 1 degree every minute. So in principle, you would need 1 hour 20 minutes to get to the -80, or one hour and so from the moment you put it there. And then you can transport this from the Mr. Frosty into the liquid nitrogen, and it stays there for many, many, many years. The moment you need it, you will simply take it out, put it in the culture dish with the right growth environment or growth media, and the cells will come back to life. Okay, so the advantages is that we can freeze the cells, we can store them, we can transport them, and we can maintain them in low passages. And that's the most important point for you. Because imagine now I have 10 plates, they are subculture number 1. Like I start a plate number 1, it's the first culture. Plating number 2, I divided it into 10 different plates. 1 of them, I kept it in cell culture, and 9 of them I stored. This is passage number 1. So I can go with my experiments up to 40 passages, and when I reach 40 passages, I say, oh, the cells are passed too many times. Let me throw this away. And I go back to the liquid nitrogen. I get another one from passage 1, and I redo the experiments again. So normally, you don't want to go more than 40 passages on your same culture. And that's why it's very important to learn how to do cryopreservation. And here there is also for you a reference guide how we do that stepwise, exactly as I said. You can also avoid having 5 different cell lines in culture. Maybe you are using 5 different cell lines today for one particular experiment, but if you keep doing this, you might mix them together. And this is an example here. Like some of the scientists, like while doing the passages, you simply mix one cell with another. And if you look under the microscope, this cell type here is totally different from that one. So you don't want that to happen. It's better than the cell line that you don't use for the next couple of months, you cryopreserve it. Okay, so let's imagine we got the cells in culture, and now we want to count them and we want to check their viability. So what we do, as I said before, let's say we collect them and we have them trypsinized after centrifugation, then we can resuspend them with tryptan blue. And tryptan blue is an exclusion marker if the cell membrane is viable, is impermeable. It's going to be telling you that this cell is alive. Where is my? So like the transparent cells, they are alive, but the cells that acquired tryptan blue are dead because most probably they have a compromised cell membrane. And so at this point, I took the cells, I trypsinized, I washed, I collected, centrifuged, and they are there. I want to know how many cells I have. So there are 2 ways to know how many cells I have there. First one is this manual hemacytometer, where you simply take very little amount of your cells and you add it to the cytometer, and you have within these squares, if you count any of these red squares, then you are counting 1 millimeter cube. And therefore, 1 millimeter cube is a dilution factor of 10,000 to become 1 ml. So if I count here 1 cell, this means I have 10,000 cells in 1 ml of my culture. If I have 2 cells in this, then I have 20,000. Like now we have, let's say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. 18, you directly multiply it by 10,000. 18 cells, so 180,000 cells that you have in the culture. So it's very easy. You go under the microscope, you avoid reading those borders outside. You make your rule that anything on the border, either I read it or I don't read it. You make your own rule on this, on estimation. Myself, I would never read those on border. I would consider them outside of the square. Some people do count them, in fact. And then the number you get here is multiplied by 10,000, and that's the total number per ml in your culture. Now, in order to get a better counting, what you should do is probably count here and there, and here and there, and then you have 4 counting, and you get the average over 4, and that's what the example I put for you. You count over the 4 red squares, and you divide that number by 4. So let's say it's 18, 17, 16, and 15. The average of those is going to be probably 16. And then you multiply by 10,000, and that's the total number of cells you have. Now, there are automated counters, and that's the reference for these, and it will tell you the total number of cells. It will tell you they are viable or not viable, because also it's depending on the tryptophan blue. Cool. I still have 8 minutes, and this is the most important slide. So is there any questions in the chat? Let me check. And this is basically exactly, that's true, Janelle. You are absolutely right. Yeah. Okay. So now, what are the types of cultures? Like, if you're doing this 1 and 2 in this figure, this is what we call the simple culture, meaning you have a cell type, one cell type, you have one growth media, and you know what are the associate needs of your plates, and you're growing these cells in the media. So that's perfect. You are now able to grow them, look them under a microscope. They are healthy. You know how many days you need to split them. Perfect. Now you're able to do the simple culture. Imagine now you want to go a little bit further, where you want to start testing different conditions. And the first thing I want to introduce to you is, like, instead of monocultures, which is one cell type and one growth condition, into a conditioned media. And in this situation, what you will do, you are asking a question: if I take a growth media from one cell type, let's say this is a leukocyte, and you're growing this leukocyte. Leukocytes are not attached. I'm just giving an example. These produce a lot of cytokines, and the cytokines are normally secreted into the media. And you want to see one of the cytokines, or what the majority of the cytokines secreted from this cell type cause for the growth or the propagation of a cancer or whatever. Then let's imagine this is the cell you are questioning. So you use that cell, and you will get the media from the other cell type. So you grow these cells, number 2. You take only the media from here, you put it on the cells that come from 1. This is called conditioned media growth. So you will now know how to how that secreted factors could affect that cell. The more complicated form of this is the use of transwells. The use of transwells. The transwells, in fact, these are wells where you can put cells growing on one end, and there is a membrane that can separate the other cell type. So in principle, those cells can migrate from the top of the transwell to the bottom. And I have the reference for you here if you want to know more about the conditions explained. So one thing you can test is the migration of this cell from here to there based on the presence of another cell. As an example, like if this is a macrophage and this is, for example, a host that is infected. Or you can test, for example, if this is a fibroblast and it's migrating toward a wound where the healing is taking place. You can also test if there is an environment where you add on this side only a cytokine and see the cells are going to follow the cytokine or not. So this can be done by transwell. And what I'm saying here is already written on the slide. This can be the migration of the cells that can be tested. So the cell migrates through the pores. On the other side, it could be chemotaxis, and it could be co-culturing, where you have more than one different type of cell. Okay, I can see that there's 5 people. Legend on the right explains it all. Awesome. Okay. I think one is bone and the other. Well, this is just, you don't have to know what is exactly the different cell types. You just need to know the application. So I didn't really emphasize on what cell we are using. Now, the transwells are considered a 3-dimensional per se, not 100% 3-dimensional cell culture, but there are people who consider them the start of the 3-dimensional. Now, if we go to that last slide, which is very important, please bear with me, don't leave the chat. So here's an explanation of the culture in the transwells. There's types where you can test the cultures under stress. Like this is a 2-dimensional, but instead of having plastic at the bottom, you have a membrane. And you can imagine if you apply suction under that membrane, you can make contraction. And this way you can make models to study heart and muscle, because these two tissues, they are under stress of contraction. So you can recapitulate this here. And finally, why to go to 3 dimensions? I wrote for you here difference between 2 and 3 dimensions. And there is also a very nice video from Corning here. Please go and watch it for 2 minutes that explains to you the different types of the 3-dimensional cultures. Now, the 3-dimensional cultures that I wanted to describe for you are the spheroids, the tissue engineering, and the organ models. The spheroids are, in fact, instead of growing the cells in 2 dimensions, like side by side, you will have the cells grown on a sphere. And this is very useful in case you want to remake organs. And that's very used in angiogenesis, because if you grow endothelial cells, in fact, in culture, you can make blood vessels. So the spheroids are very used in that type of research. Also, it's very important for the angiogenesis processes, that because cancer needs the generation of new blood vessels. Therefore, if you block a blood vessel formation, you can block cancer progression. And there's a lot of research taking place on spheroids in order to block the cancers from growing into having ability to access the blood vessels and form new blood vessels. The other one is the tissue engineering, where you will take the cells and grow them in culture. And each of these 3 that has a video, please go and watch it. It's very interesting. Where imagine somebody had a wound from fire on the skin or injury, where you take the keratinocyte and fibroblast and you grow them and you make patches, and you can come back and use the own cells to patch on the patients. This is also used in the heart and valve, where we engineer cells and we use them on matrices on the valves, as well as in organs where there's no regeneration, like bone models and cartilages. And finally, the organ model, these are organoids. So it's very close to be an organ that is made in vitro, in a dish, in a culture. And you can use it to test for drugs. Like imagine you have a new drug for liver, you can go and test that drug in a situation, not only on the cells of liver in 2 dimensions, but it's all over like an organ in 3 dimensions. Please go and watch these videos. And here, this is a list for you with the types of cells that we normally use in the lab. That's the 3T3 cells here. There's the HEC in the previous slide, 293 cells, HEC. There's the C2, C12 from the muscle. And the most cells we get is from the company called ATCC. It's the greatest, how to say, portal for buying any cell type. And there's a guide for you if you would like to read more. And with this, I think we are 1 minute after. If you have any questions, please let me know before you leave. Otherwise, we can carry on next Thursday with the fractionation of cells.
Thank you.
You are welcome.
Thank you, Professor.
You're welcome. You're welcome. Please, if you have any speculations and questions, if you don't want to do it now, you're welcome to keep it for next session.
Professor, I had a question not related to the lecture, but about the first assignment.
Mm-hmm.
Do you know when it will be posted on Brightspace for us to see?
Tomorrow morning, in fact, I have the meeting with my TAs at 10, and it should be uploaded tomorrow at 10. And since you asked, I don't know who is talking with me because the slide is covering the
My name's Chelsea.
Chelsea. Okay, Chelsea. So I will go back, in fact, to the slide where the assignment comes from. And
Okay.
what I was thinking is to explain it for you guys in the next session. But since you already asked, I will quickly. Okay, so in principle, the first assignment is going to be based on this slide. I will provide you with a scenario where you have to trypsinize the cells, count the cells, and tell me what is the exact number of cells at the end. There are a little bit more difficult than just counting and multiplying by 10,000 because the culture might be diluted and stuff like that. So this is the first assignment you will have due by next Thursday, not next Thursday, the Thursday after. So it's gonna be very, very easy, few minutes, and it will be done.
Okay, great. Thank you.
You're welcome. So not related to the lecture, but I was wondering if the assignment was posted. No, I just answered this question. So is the assignment due September on Brightspace? Yes, and as I said, it's gonna take you minutes to solve. Just to clarify, tissue engineering is used for models for organ with limited or no region. Well, not necessarily only for those. There's a lot of complications with tissue engineering. We can discuss it if you want next time. When you will be making a notes of an examination for a student registered with SAS. Okay, so I can't say your name for the confidentiality, but since we are having everything online, I would like to know from the department what should we what are the requirements to add for you the notice. And this depends on you. I will be emailing you tomorrow after the meeting with my TAs. If you are willing to perform the exam online, or you would like to do the exam in the exam center. So if you are doing the exam online, there's no need for the notice because it's going to be all managed by me and the TAs. And then we will just consider what you required. You're going to be at home or whatever you prefer to be and perform the exam. Now, if you are requesting to be in the exam center because you think that you have less visual and audio distraction, then it's your choice. And in that case, I have to send the notice, and I have 10 days before the exam to do that.
Excuse me, Professor. I just have a question about the lecture slides.
Yes.
So I wasn't able to make it for the first class, but to clarify, for the midterm and the final, is good like a good way to study for those exams to go through the lectures as well as the notes that we make during the class and just study off the lecture slides, or is there any additional information that we should be looking at?
Normally, the majority of the questions will come from the slides and the information in the slides.
Okay.
So if I, for example, give you a reference, and if you go back to this lecture today, you can see that there's a lot of references, but these references would be like few pages.
Yeah.
Or if there's a book like 500 pages, I also write down page 161 to page 163.
Right,
This way, I want to really gauge your your attention to only a specific number of pages.
Mhm.
And I also try to use the videos for you so that you have the visual memory.
Yeah.
In case. Other than that, you don't need to go and read other references.
All right, sounds good. So
Okay.
the lectures are pretty
Lectures should be more than enough for you.
yeah, okay,
Yeah.
perfect. Sounds good. Thank you. It's nice meeting you.
Thank you very much. Have a good day. Nice
You as
meeting
well.
you. Nice meeting you too. Any other questions before take off? Okay, thank you very much all.
Have a good day, Professor.
Thank you. Have a good afternoon. In the syllabus, it says the assignment is due 17. So any questions more on the yes. So there was, I'm not sure if you are still with me here, but there was a question if the recordings will be posted. Yes, the way it works with TLSS is that you will, after this Zoom meeting ends, it's gonna be processed online on somewhere on a OneDrive thing. And because of the space issues, we have a link for you. You can go to that link anytime and watch the recording. So I will be emailing you with that link every lecture. After the lecture is ended,